My talk is about using spectral information in agriculture and plant science. And before we start, the question is, what is the development of agriculture? And the problem in agriculture is that the area where we can grow food is decreasing from year to year, and the people that are fed by a farmer increase each year. So where we had in the 1900s, we had 4 people fed by one farmer. It was in the 1950s, we had 10 people, and today we have more than 100 people. And how is this possible? This is only possible by improving technology and organization. So we are talking about optical sensors here. So what are optical sensors used in agriculture? They are used for 2 different things. The first thing is field monitoring to track growth and development of the plants. The second is to identify development stages, biomass, and yield potential. The second topic is detection of, for example, stress. There we have abiotic stress, like drought stress or nutrient deficiency. And the second is biotic stress, like diseases, insects, or wheat. And optical sensors are used because they are objective, they give you reproducible results, and they are highly automated, and they enable to measure with high throughput. And for all the people who are familiar with hyperspectral imaging, there are different things that influence the hyperspectral data cube you get in the end. So one big thing is the experimental setup. For example, the distance between camera and the object of interest. Another topic is the sensor characteristic, where you have the dark current, the sensor noise, the distortion of the camera, for example, smiling and keystone effects. And of course, the illumination, so the spectrum of your light source and the change of it over time, or scattered light. And now there comes another class of influences when you try to measure plants, because plants are not very easy to measure. So you have, for example, different leaves that look similar, but they have a different spectrum. You have inclination influence by geometry. You have absorption of the plant tissue, transmission and backscattering, or, for example, effects due to growth. So how is a typical plant data cube looking like? On the left, we have a typical hyperspectral data cube consisting out of 2 spatial and 1 spectral axis. And on the right side, you see how a typical plant spectrum is looking like. So you have, by the way, do you see my mouse pointer here?
We see the mouse pointer, yeah, okay. But you didn't activate the laser point mode in PowerPoint.
Ah, okay. How can I do this?
We only see the mouse cursor.
Okay, now it's okay.
Yes.
So we have the peak. We have the peak here between 500 and 600 nanometers. Before it's very low, and then we have a valley here between 600 and 700 nanometers, and then it goes up to a plateau with a very high reflectance in the NIR area. So this here is a very typical plant signature. So, but why is it looking like this? It looks like this because we have in the area before 500 nanometers, we have the absorption due to carotenoids. Then we have the chlorophyll reflectance at 500 to 600 nanometers, and the absorption of chlorophyll in the area between 600 and 700 nanometers. And then we have the NIR peak in the plateau. And this is why plants are looking green, because they absorbed, but in the green area, they reflect. Okay, so what are we using in agriculture to get multi or hyperspectral images? So when we talk about multispectral in this field, we talk about, we have a camera that is able to measure the NIR plus the visible area, and it uses up to 20 channels. Everything below 20 channels we define as multispectral. And one camera that is giving you a good multispectral image is the Megasense Red Edge, for example. It has 5 channels, blue, green, red, and NIR. and you can use it to get a 3 megapixel image with 5 channels. If we talk about hyperspectral imaging, we talk about more than 20 channels, and one very common example is the Spectral IQ camera, which has 200 channels and measures in the area of 380 to 1000 nanometers. So these are two very common cameras for using in agriculture. So where do we measure? We measure indoor, of course, and outdoor. If we measure indoor, you mostly have a line stage with the camera and the light together, and then we measure from the cell to the single plant size. So that means we measure, for example, disease symptoms, single leaves, or a single plant. If we go outside, we measure everything from the single plant to a smaller plot to the big field. And if you go outside, you have to transfer your line stage somehow to the field, and this means you need some kind of shadowing. This is why you see this tent here, because we put our lamps outside. We want to reduce the sunlight, and then we need something to move our cameras over the field. This is why you see these constructions here, often combined with a bike. So you have what we call photo bike. So you have your cameras moved over the field. Another option is to use drones as a carrier platform and to fly with your camera over your plants. So one thing which is very important in agriculture and is the vegetation index. A vegetation index is, in this case, I want to explain the NDVI index, the normalized difference vegetation index. And this index takes two wavelength areas and it combines these two. And in this case, we take the red area here and the NIR area here. And if we have a healthy plant, we have a high reflectance in the NIR and a low reflectance in the red area. And this together gives a high NDVI value. And this gives us the information that the plants are very healthy. If we have a stressed plant, this reflectance is reduced. So we have a reduced reflectance in the NIR area and a better one in the red area. Then we get a lower value here. So we know that something has happened to our plant and it's somehow stressed. We don't know what, but we know it is stressed. So we can also transfer this to the field. So when we fly over our field and we use a spectral camera below, and then we can, of course, get the RGB image of our field, or we can get the NDVI image of our field. And then we see that there are some plots that are, let's say, looking different among each other, and then we know that something happened to our field, and then we have to go there or to start our analysis directly at these points that look not so healthy. So if we talk about hyperspectral traits, I know vegetation index is very, very basic. So in this graphic should show you that there are different levels of hyperspectral traits. And they all, let's say, they differ in the complexity of the data processing you need to reach them. So trait level 1 means that we have a very basic analysis of the spectrum. We have, for example, the canopy spectrum, which means one measurement for the complete plant. This could also be a vegetation index. So if we move to trait level 2, we have to segment our plant into the single organs, for example, the leaves. Then you get information per leaf. And then you get, for example, the spectrum of a single spot of a disease, or the spectrum of a single leaf itself. And if we move to trait level 3, it means the complexity is increasing. We measure over time. So we measure each day or each week, however you want it. And then we can describe the spectral development of the plant spectrum. or for example, the spectral development of the vegetation index. And trait level 4 means we need a further sensor that supports our hyperspectral sensor, for example, to reduce the effects of the geometry or the inclination. And then you can correct for the geometric influence, for example. And this means you have a multi-sensor setup. So these are the very common categories of hyperspectral traits. And how is a typical hyperspectral workflow looking like? So we have in in this case, I want to show you the workflow for detection of disease infection in the field. So we have the experiment setup, we have the data acquisition, maybe over time. We have the data normalizing, the filtering, and the calculation of the region of interest. Then we start the machine learning, where we want to differentiate between the different treatments. And if we can do this, the next question is, do we really need hyperspectral, or can we switch to a multispectral approach? And then you have to calculate an importance for the different wavelength, because if you want, what you want in the end is you want to use a multispectral camera in the field instead of a hyperspectral camera, and reduce the amount of work you have. And if you know which are the important wavelengths, you have to combine this technical knowledge with the biological knowledge. And then you know why you need just these, let's say, 4 or 5 wavelengths in the end. So this here shows you very in detail how the single steps for the workflow are looking like. But I want to be very rough here. The idea is you start with images, you image your plant, then you switch the images to a real data set where you reduce the noise and you separate the foreground background and you enhance the quality of your data. And then you start preparing it for machine learning. Then you have information, and then you know about how to interpret your data, what you need to identify, for example, a disease infection. And in the end, we want to go to knowledge. And in the end, this completely is the workflow you have to cover. What does it mean for our workflow I showed you before? When we want to, let's say, I have an experiment with control and effective plots. I use machine learning for classification of, or to separate between control and effective plots. I start the feature selection, and then I get an importance for each wavelength. And then I have to answer the questions, why are these, let's say, peaks important for my classification? And this is not very easy, because normally the people dealing with the machine learning are specialists for machine learning and not for biology. And this is why the workflow for detection of diseases in the field needs different specialists. You need a specialist for experimental design, you need a sensor technician, you need a computer scientist or a machine learning expert. In the end, you need a biologist. Because this, what I show here, is not a job for one profession. This here is important because you need a multidisciplinary team. So people of different professions, different ideas have to work together here. So now I want to show you 3 different, let's say, scientific use cases, because these are use cases of my daily work. The first one is an application where we have the disease detection in the greenhouse. And here we used sugar beet and an infection with Rhizoctonia solani, and we used the Vimeo camera measuring it from 380 to 1000 nanometers, and we extracted the spectrum of each plant. And we had 3 different classes here. We had one class with the control, which was healthy, one class with a low infection, and one class with a high infection. And we used machine learning, in this case, support vector machine modeling, to classify just using the spectrum if we can identify if a plant is infected. And the result here was that we were able to detect an infection 3 days before a human being can detect the disease with the eye. And this is for the greenhouse, which means maybe we are in the field a lot faster. The second application is the transport of this to the field. And here we used sugar beet and in inoculation with Cercospora verticula. And we used a Vimeo camera, the same as before, but on a tripod. And we tried to use vegetation indices. You see that on the right side, the healthy one is the upper one. And you see that the vegetation index is looking completely different from the infected plot. And so this means you can use a simple vegetation index to differentiate between these two groups. Okay, and the third application is the prediction of yield in open fields. Here we used the Sentinel-2 satellite data, and especially the NDVI coming from the satellite. And we combined it with neural nets for prediction. And then our target variable was the yield that was measured by a state-of-the-art harvester in the field. And then we tried to model a neural net that was able, just by using the NDVI data from the satellite, to predict the yield of the field. And we are very good in this. And this is the highest, let's say, roughest scale we used during my work, the satellite data. So what is the next step? The next step is shown here. On the left side, you see the NDVI values of a sugar beet. And you see that we have some highlights here, especially where we have a big inclination, and this is shown here. The inclination of this sugar beet is very similar to the NDVI image. And that, what I want to show you here is that we have a big correlation between the NDVI and the inclination map. And this means somehow we have to combine the 3D information of our plant, the sugar beet, and the hyperspectral image, so that we can correct for this inclination influence, so that we in the end have a 3D point cloud of our plant, where I can extract at each point the spectrum. And this was done here for a sugar beet in the laboratory. So this is the next and the further step, what I would say is cutting edge at the moment. Okay, so what are the challenges, especially in the field context? The challenges are sensor-based, the trade-off between distance and resolution. Especially, I cannot detect diseases from the satellite, but from the drones in each case. So what is really the scale I need? What are the right channels and which are the important ones? Or if we talk about the experiment, in the field experiment, do I really measure my disease or a nutrient deficiency or maybe a mixed infection? How can I be sure that what I measure is disease? And who depicts the ground truth? Just the humans, or can I get anything else? If you switch to the analysis, the question is, how can I process huge data cubes in high speed? And if I have a method that is working and I have a second one that works too, which of these methods gives me the most valid results? So this is a really big challenge. So this is the future of agriculture, and the main point today, or in my talk, is that technical progress has enormous influence on agriculture as we know it today. And what you really need is interdisciplinary research. So people from different professions that work together with ideas, and the big task for the future is to combine all these together to one big solution. Yeah, thank you.